All right, hello everyone. We are here this time to talk about fish. Not the swimming kind, but FISH, fluorescence in situ hybridization. And so you can use it to label fixed um, RNA sequences within basically any tissue you can get your hands on. So, like a mouse embryo, fly embryo. This is actually a zebrafish embryo, xenopus embryo, or tissue section on a slide. You could even do it with cells, I suppose, if you wanted. And so what you're going to be looking at in specific is going to be a stain of whatever tissue or embryo, whole mount, if you do a whole mount. Well, anyway, you're going to be looking at the staining of the whole thing, but if you were able to zoom in really, really far and see individual molecules, basically what you'd be seeing is your fish probes attaching to RNA. So let's look more at what that entails. Oop. So, oh no, let me put this right here. All right, so what you're going to be actually seeing is your labeled RNA. So when transcription is occurring, right, you've got RNA polymerase running along the DNA and writing the RNA, right, the mRNA. Well, what will be soon mRNA. So it's writing the transcript. And the transcript is going to include exons, which are pink in this imaginary hypothetical, and introns. So exons pink, introns yellow. And so sometimes you want to be able to label the introns. So you would make probes that are complementary to the intron. And sometimes you would want to be able to label only mRNA, so RNA that's already been spliced. So your introns are cut out. And if you want to label both, you can actually make probes that are that are inside of only one exon. And if you want to label only mRNA, then you would make a probe that spans this splice gap, right? So if you make a longer probe, it's going to preferentially bind to the mRNA's post-splicing, right? Or if you want to label active transcription, or as close as possible with this method, you would make intronic probes, right? So they're complementary to the introns, and since splicing occurs pretty rapidly after the transcript is formed, but the mRNA hangs around quite a while after, if you make a probe that's complementary to part of an intron, then you can often actually see the active transcription as it was occurring at the time of fixation, right? And so then you want to be able to see these probes in your actual samples, right? So you need to label them somewhere. So as you can see with these probes here, you've got these little green blobs, right, which I've labeled hapten. Hapten is just an antibody target, right? So it's a label that's actually put onto the probes. And I've got a little thing over here, if I can just scroll up a little bit and pull it into view. So the way that the probes are made is you take a complementary DNA sequence. So you basically take the DNA sequence that you want of the RNA that you want to label, and you flip it around, and then you put an RNA polymerase promoter, so like a T7 promoter or an sp6 promoter or T3 promoter. There are several different RNA polymerases, and then you do in vitro transcription of that complementary DNA to get the complementary RNA. And to put the label in, what you actually use is a conjugated UTP. That's what is generally used. Technically, you could, put that conjugated label on any of the other ones, but as a rule, it's UTP because that's not going to be because it's unique to RNA versus DNA, right? And anyway, so then you do that and you get your probe, which then has the conjugated label written into the transcript of your RNA probe. And the RNA probe itself is made of RNA, right? So then you do an in vitro hybridization. So you get your RNA probe to stick to your RNA target. Let me pull that back into view for you. Okay, and then in order to be able to see it, you still can't necessarily see it because your label is not necessarily very bright, or maybe it's not fluorescent at all, and so you want to be able to, and also you're not going to be able to make a very stable fluorescent probe, even if you do use something like FIT-C, right? The process of making these probes and then storing them is not going to be ideal. Whereas if you order like a secondary antibody of the Alexa series, for example, then you're going to have a lot more stable fluorophore that's going to be a lot brighter and a lot easier to see. So what you generally do is you make, you purchase, let me pull this to the front, bring to front. You purchase an antibody that's made in, let's say, for example, a goat, and the antibody is to that label. So then your antibodies bind here to your labels, which are bound to your RNA sequence target, right? And then you would, after doing an antibody staining of, so you do the in situ hybridization to get the probes bound, and then you do an IF basically. So first step is you use your antibody, which is anti-label that's on your probe, and then you would do a secondary antibody staining. This one is also in the back, so bring different. And your secondary antibody is to the goat antibodies, right? In this example, so like an anti-goat Alexa 555. So you would get then a red fluorophore. So you'd get red fluorescence labeling your probe, and then you're able to see it. And I will put an example. I'll put several examples of fish stainings in the thumbnail, so you can see what that looks like if you don't know. But that is the basics of how fish works. So, happy sciencing. Talk to you later.